Hello, am I audible?
Yes, ma'am, you are audible.
Yes, ma'am.
Shalja madam, there is a request. What we can do is when the talk will be starting, we can mute all others except Dr. Jitender so that the talk can go unhindered.
Actually, two streams are there, no sir. There is somewhere two streams on, sir. That is the problem actually. YouTube and Zoom in one system means resound is coming. Now we stopped, I think.
Yeah, that will be fine. No? That will be. As long as it doesn't disturb the talk, we can go ahead.
Shailaja, am I audible?
Yes, you are audible, Padma.
Sir, can we start?
Yeah. About 48 participants have joined, so we can start.
Very good afternoon to all the participants. Now I welcome Dr. Shailaja to give the welcome.
Good afternoon to all. As part of the Azad Ka Amrit Mahotsav 75th year celebration of independence, IARR in association with Society for Advancement of Rice Research organizing a series of webinars. Today's webinar is on the interesting topic low P tolerance in rice, mechanisms and way forward. Hope this is very useful to the rice community. My hearty welcome to our beloved director, Dr. R.M. Sundaram, who is the driving force behind all these programs. My cordial welcome to today's chief guest, speaker of the webinar, Dr. Jitender Giri, NIPGR New Delhi. Sir, thank you very much for accepting the invitation, sir. I extend my warm welcome to executive members and live members of SAR, heads of the sections of IARR, my colleague scientists, distinguished delegates. Once again, I welcome all of you to the interesting webinar. I thank our director for giving me the opportunity. With this, I request our director to deliver the opening remarks.
Dear Dr. Shailaja, Dr. Padma Kumari, Dr. Satyendra, and also Dr. C.H. Padmavati, dear Dr. Jitender Giri, heads of sections, my dear colleagues, ACRIP partners, and also students and research scholars working at IARR. I'm delighted to have Dr. Jitender Giri, who's one of the best young scientists in the country, I would say in the field of nutrient use efficiency. And he also happens to be a good friend of IARR in the sense that we have been having collaboration for the last almost 3-4 years, and the progress has been outstanding. And we have been collaborating on two aspects, that is tolerance to low soil phosphorus and then phosphorus use efficiency. And I understand that this nutrient use efficiency, or particularly pertaining to phosphorus, is very important in the coming days because the phosphorus or phosphate reserves are very finite, and the cost of phosphatic fertilizers are also increasing year after year. So in this context, minimizing the input application and maximizing the benefits, or shortly developing varieties for low phosphorus tolerance, is the mantra for with regard to what you call as sustainable agriculture. And Dr. Jitender Giri has been working on some of the basic molecular, biochemical, and then physiological aspects related to this topic. He's also working along with us, and then we had a significant headway in terms of understanding some novel low soil phosphorus pathways associated in many varieties which don't have the popular QTL PUP1. And Dr. Jitender, many thanks for accepting our request for this particular prestigious webinar. We have this webinar series once in a month wherein we invite some of the best in the field or best in their particular target area of field, and then request them to deliver a lecture for not only enlightening our own staff, but also the staff of nearby institutes in ICR, and also the staff who are part of our All India Coordinated Rice Improvement Project Network. I now request Dr. Satyendra, who is also a close friend of Dr. Jitender, to introduce the speaker.
Thank you, sir.
as I think Sir has already given a brief introduction of Dr. Giri, and he is friend of mine and also collaborator with IIRR, and I think many of us, we know about him. He's a very bright and known scientist in the rice community. For the benefit of the members, those who are not from this field, I will read out his brief CV. He is a very well accomplished scientist in India. He is scientist at National Institute of Plant Genome Research, New Delhi. He did PhD in molecular biology in the year 2009 at University of Delhi South Campus. It was the same time that I was doing PhD in ICAR-IIRR. So, and his research areas includes plant nutrition, molecular biology, biotechnology, genomics, environmental biotechnology. Specifically, his work is very well cited and very well known in the area of low P stress tolerance and also phosphorus use efficiency. His landmark papers and very well cited papers are there in this field. He has research experience of 15 years behind his career. He has been abroad as a visiting scientist at Penn State University, USA, and also at University of Nottingham, UK. He has published more than 43 research papers in top national and international journals that includes Nature Communications, Plant Physiology, Molecular Plant, New Phytologist, and also Plant Biotechnology Journal, and many more peer-reviewed research journals. To credit the awards that he has got for his outstanding work, he has been recently in this year he has been awarded as the Fellow of the National Academy of Sciences India, NASI. He's the first plant scientist to win highly prestigious Swarna Jayanti Fellowship Award in the year 2018-19. Beside this, he has got NASI Scopus Young Scientist Award, IYBA 2016 from DBT, INSA Young Scientist Medal, NASI Young Scientist Platinum Jubilee Award, and also DBT Crest Fellowship Award, along with several other awards that I think that it's difficult to read out all the names. So I am just giving most significant ones. And also he has been invited as the lead speaker, also keynote speakers in some of the very well-known national and international conferences. So this is a brief introduction, and with this brief introduction, I welcome Dr. Jitendra. Thank you. Sir.
Yeah, for this session, Dr. A.P. Padmakumari will serve as the moderator. Dr. Satyendra and Dr. Shruti will record the proceedings. So now I now invite Dr. Giri to start his lecture. Sir, we have at least 57 participants, and many have also joined through our YouTube medium. Sir, the time is yours, and kindly start your lecture, Dr. Jitendra.
Thank you, sir. Thank you, sir, for invitation, and thank you, Satyendra, for kind words. That was not a brief introduction. That was a heavy introduction. In fact, you made me conscious after listening to those things. But anyway, yeah, so thanks again to organizer for having me here. It is always a pleasure and at the same time challenging to talk to you guys. So pleasure because you rarely get opportunity to speak exclusively to rice researchers, and I believe that everybody sitting here is actually working on rice. So there is so much give and take, so much to learn. And for the same reasons, it is also very challenging for a person like me who are actually lab rat. We actually do not do any work in the field. So we have least knowledge how actually plant is behaving in the field. And I think this is where we thought of this collaboration, and thanks to Dr. Sundaram, his untiring efforts that that collaboration was possible and we could get that project. And I'm sure we will make something good out of it, and which will also lay the foundation for our second stage collaborations in many other areas. So thank you very much once again. I'll start. I'll take about 45 to 50 minutes. I think it's okay.
Yes, sir. Yes.
All right.
That'll be fine, Dr. Jitendra.
Fine, sir.
Time is all yours.
Yeah. I'll try to be brief. I hope you can see my slides.
We are able to see, Dr. Jitendra.
And they are moving?
Yeah, they are moving.
Perfect. Thank you, sir. All right. So in next about 45 to 50 minutes, I'm going to talk about low P tolerance in rice, mechanisms, and way forward. When I say low P, phosphate starvation or phosphate deficiency, these terms essentially mean same thing. That means the phosphate deficiency either in the soil or in the media where we are growing in the growing plants. I'll start with some basics on phosphate. I'm sure this audience doesn't need that much introduction on phosphate. Then I'll discuss about the molecular basis of the mechanisms which plants employ to cope up with the phosphate deficiency, which I would follow with some of our examples, how we are trying to improve phosphate use efficiency in rice before we discuss the way forward to address this problem. I will quickly introduce the phosphate problem here. Plants or no organism can survive without phosphate. It's a part of DNA, RNA, ATP, and membrane in the form of phospholipids. So nothing can survive. And plants, you all know that plants get phosphate from soil, where it is a limiting factor for plant growth because phosphate is available in really low concentration because of high interaction with the within the soil. It interacts with the calcium, iron, aluminum, and form inorganic insoluble complexes, or almost 50% of phosphate is present in the organic forms. So plants, despite having phosphorus, they face phosphorus deficiency because roots can absorb only inorganic form of phosphorus, which is H2PO4, which is a soluble form of phosphorus. So this form is present in a very less amount in soil solution, hardly from 2 to 20 micromolar, and which necessitate the use of phosphate fertilizers in the agriculture. And source of such fertilizers are actually non-renewable rock phosphate, which is highly asymmetrically distributed in the world. Morocco literally has all the phosphate, and our situation is pretty bad. India's total reserves are about 200 million metric ton, and these are actually poor quality phosphate ores. And the world requirement for 1 year is 191 metric ton per year. So in a way, you can say that India's total phosphate deposit can hardly meet the 1 year phosphate demand of the world. And this makes us the largest importer of phosphate fertilizers. Almost 50% of phosphate import is for India only, and majority of it is going in agriculture. So in a way, you can say, these are fertilizers are really expensive. Then subsidy comes in, and probably 2 years back, the fertilizer subsidy budget was more than 1 lakh crore, which was higher than our union health budget. So you can see the importance of these fertilizers, without which we really can't even think of having the food security. So the supply of phosphate fertilizers is not only an economic issue, it is also a strategic issue for a country like India, where we are entirely dependent on import. So there are two problems. One is that phosphate has high fixation rate in soil. Second is that we do not have phosphate supply. And then there is a third problem. The crop like rice, which is the largest consumer of phosphate fertilizers, has a very poor phosphate use efficiency. Rice has about 20% phosphate use efficiency. And this picture, you can see these are from your own institute, thanks to Sundaram sir. So rice variety is growing in phosphate fertilized field, and here is the phosphate deficient soil. Although you will never see such kind of deficiency in farmers field, these are the experimental plots in ICAR-IIRR. But in farmers field, there is a considerable reduction in the crop yield in the event of phosphate deficiency if that is not compensated with the application of phosphate fertilizers. So in soil, phosphate is mainly present in this form, this green one form. These are the inorganic soluble forms. But this form is present in the least amount, and this is the only form which plant can absorb and assimilate. Other forms are bound forms like aluminum phosphate, iron phosphate in acidic soil, calcium phosphate and magnesium phosphate in alkaline soil. 50% of soil phosphate is also present in the form of organic phosphate, that is in the form of phospholipids, nucleic acid, which is coming from decaying biomaterial. They could be plant residues or they could be dying microbial diversity in this soil. So these forms, red highlighted forms, these forms have to be hydrolyzed first, to mineralize first, then only plants can use phosphate from them. So majority of the efforts, they are targeted towards enhancing the uptake of this free form so that plant can absorb phosphate before it gets fixed into these forms, or other strategies they follow where they want to target the enhanced solubilization of these fixed forms. So how actually plants behave under phosphate deficiency? So plants employ a wide array of adaptation tactics. These adaptation tactics actually starts with the activation of several genes, which we call as the phosphate starvation induced genes or PSI genes. These genes then regulate other pathways for phosphate uptake. For example, lateral root length is increased, and there are other root architectural modification in the plant where either the root hair length is increased, root number is increased. So these adaptations are targeted to increase the root surface area for phosphate uptake. In some plants, the roots branch extensively and form a kind of a cluster root, especially in the plant like white lupin. And in that cluster, highly branched roots secrete acid phosphatases and organic acid to solubilize the bound phosphorus. Some plants, including rice, they also secrete organic acids, many carboxylic acid in the soil through root, where and this organic acids, they can hydrolyze those inorganic bound forms like aluminum phosphate and iron phosphate. So if you see all these forms, all these adaptations from here upward, they are targeted towards the enhanced phosphate uptake. But then there are also adaptations which are targeted towards efficient use of absorbed phosphate. And here I'm highlighting one such adaptation, which is membrane remodeling, where plants try to extract phosphate from membranes, membrane phospholipids, and those lipids can be replaced by other phosphate-free lipids. So plants use both strategies. Plants, on one hand, enhance the phosphate uptake from the soil. At the other hand, plants try to maximize the use of cellular phosphate. Now, this phosphate is taken up by phosphate transporters in the plant, and there are a variety of phosphate transporters. For example, PHT1 class transporters, which are plasma membrane localized transporters, they bring phosphate inside the root. Once it is inside the root, another class of phosphate transporters, which is 4-1, it loads the phosphate from root into the xylem, and from xylem it can easily go to the different organs. And then we have organ specific phosphate transporters, variety of them, which ensures the partitioning of phosphate to the priority organs and also help during the senescence. So there is a network of phosphate transporters from uptake to the distribution in the plant. Now, you will be surprised to know that majority of these phosphate starvation induced genes, they are regulated by a single transcription factor. In fact, many phosphate related adaptations, they are regulated by the single transcription factor, which is PHR2 in PHR1 in Arabidopsis, and its homologue PHR2 in rice. So these transcription factors, when plants face the phosphate deficiency, it binds with the promoter region of many of the phosphate starvation induced genes by binding at the cis element, P1BS element, and activates the expression of downstream genes. What happens in the normal condition? In normal condition, in order to restrict the phosphate over accumulation, which is actually toxic to the plant or detrimental to the plant, there is another protein which is SPX1. This SPX protein interacts with PHR transcription factor and does not allow it to bind with the promoter region of the target gene. So their transcription is not activated. So there's a regulation inside the plant to enhance the phosphate uptake, at the same time to prevent the overaccumulation of phosphate in the plant. Now there is additional level of regulation of this PHR transcription factor. SPX1, while inhibits its interaction with the promoter of the target gene, another protein SPX4, it binds with the PHR in the cytosol and it doesn't allow its translocation into the nucleus. So when SPX4 is there, and another thing which I'm going to talk to right now, when they are there, they does not allow for the PHR to go into the nucleus, and this happens under the high phosphate condition. In low phosphate condition, SPX4 is degraded, and now PHR1 can enter in the nucleus, where SPX1 cannot bind with the PHR1, and transcription of those responsive genes can be activated. Now here's an interesting thing, that what is the sensor for phosphate levels? How plants sense that now this is the stage where I'm facing phosphate deficiency and now I'm not facing phosphate deficiency. So how plants actually sense it? Now, interestingly, this is not sensed by the level of free phosphate, which was believed for very long years that actually phosphate, soluble phosphate level is the measure, and it is actually a sensing molecule, which is sensed by plants. Rather, it is inositol phosphates, different species of inositol phosphate, and it is IP8, inositol phosphate 8, which is used as a proxy to sense the phosphate level. So what happens under high cellular phosphate condition? There is plenty of inositol phosphate 8, and this IP8 actually works as a glue. And while working as a glue, it binds the SPX with PHR1 or SPX4 with PHR1. So when this glue is there, PHR1 is caught either in the cytosol or in the nucleus, and there is no transcription of phosphate starvation responsive gene. What happened under low phosphate condition? IP8 is hydrolyzed into IP7 and subsequently into IP6 and releases the inorganic phosphate, which is used in the cytosol. Now, since IP8 levels are low or IP8 is missing, that glue is missing. Now, since that glue is missing, SPX proteins, they cannot bind with PHR, and therefore they cannot suppress the phosphate starvation response. So this is an interesting adaptation in the plant. And in fact, this scheme of regulation of phosphate starvation response at molecular level is discovering, day by day we are discovering it in many plants, and more or less this scheme is conserved. Many of the homologs of these genes, they are doing essentially same functions in different plants, be it monocot and dicot, crop plant or wild plant. So this scheme is very much conserved for molecular regulation of phosphate starvation response in rice. Now, with this basics understanding how plants actually sense and respond to phosphate deficiency, if you talk about the strategies for improving phosphate use efficiency in plants, we can broadly classify them into two broader group. One is the phosphate acquisition efficiency, that means the efficient phosphate uptake from soil. And second is phosphate utilization efficiency, and I call this as an internal phosphate utilization efficiency, which means the efficient use of phosphate which is already inside the plant. It could be partitioning or it could be the solubilization of organic phosphate. And this is my third talk in IIR, and I made sure that this talk should not overlap with previous two talks. So here I'm going to show you two examples, one about the PAE, which is the acquisition, where I'll show you example how altering root system architecture we can improve the phosphate acquisition. And then I'll give you a second example, which is about internal PUE, where I'll show you our data, how membrane lipid remodeling can be targeted for improving phosphate utilization. All right. So my lab is working for about 10, 12 years for improving phosphate use efficiency in rice. And we actually started with two varieties, Dular, which was relatively tolerant to phosphate deficiency, and PB1, which was relatively sensitive to phosphate deficiency. And by generating a huge amount of comparative transcriptomic data combined with the morphophysiological traits, we actually identified few genes which can be targeted for improving phosphate uptake or its use efficiency in rice. And we have demonstrated using transgenic approach a couple of them in rice. I can show you one example. Here, this PAP21B, it is an acid phosphatase, purple acid phosphatase. And this enzyme can release phosphate from variety of organic phosphate substrates, be it DNA, ADP, ATP, almost 15 substrates we have tested, and this enzyme can release phosphate from them. So we proved that in normal wild type plant, this protein is actually secreted in the soil by roots. And in soil, it can hydrolyze the organic phosphate compounds, which we demonstrated in these transgenic lines. These right side 3 transgenic lines are the PAP21B overexpression lines, and here is wild type. And in these plants, we did not use any chemical fertilizer at all. We simply supplied the normal field soil with the cow dung. Cow dung was the source of organic phosphate. And if you can see, the growth in overexpression lines is better, and these lines also accumulate and more phosphate. And since we did not supply any phosphate from outside, that means these plants were able to extract more phosphate from the organic cow dung. So this is the one example where you can alter the root functions, for example, secretion of acid phosphatase here by roots, and you can improve the phosphate utilization and uptake efficiency. Roots are, because of this reason and because of several other reasons, roots are now being termed as the key for second green revolution, because first green revolution was targeted to upper ground part, and now many experts are believing that next green revolution has to be targeted to below ground part, because below ground part can help crops which can grow with less water and less fertilizers in marginal soil. And this Drovan example, I'm sure all of you know about this example, that this is a one protein which is an auxin signaling related protein. And when this gene is present or this allele is present, the plants, they develop a deep root system. And when this allele is absent, the plant, they develop a shallow root system. And this was the impact of change in root growth angle at the early seedling stage. If the angle is quite close to the surface of the soil, the roots remain close to the soil surface. But if angle of the growing seedling root of growing seedling is steep, the mature root system develop a deep root system, and this plant become a drought tolerance because water availability increases with the soil depth. So slight change in root angle can improve the drought stress tolerance in rice. And this PUP1 example, again, you people already know this example. This is the only QTL which has been cloned and characterized for phosphate deficiency tolerance. And this gene encodes a receptor like cytosolic kinase, which results in the more root number at the early seedling stage. And at the mature plant, if you see in the low phosphate conditions, the PUP1 carrying plant has more roots, a vigorous root system that results in the more phosphate uptake. And this PUP1 QTL has been in progress in several low P sensitive rice. I think one example is itself from Dr. Sundaram sir lab, where they have transferred it to improved Samba Masuri 1, and it is improving the phosphate uptake efficiency. But remember, this PUP1 QTL is targeted for uptake, not about the use efficiency. But for example, in PUP1, we are having a bigger root system or a vigorous root system. But we should remember one thing, that roots are non-photosynthetic organ, and therefore they are dependent on the supply of carbon from shoot part. So when root is giving back water and nutrient to shoot part, root is consuming a lot of carbon, which is coming from a shoot part. And sometimes this cost becomes so high that it may even impact the shoot growth. Although this is not the ideal picture, this I have used only for representation. But remember this thing, that an efficient root system is the one which put the minimum carbon cost on the shoot part. And those roots, we call them as a smart roots or climate or the or the adapted roots. So, but we do not understand the what kind of root architecture we need for a different soil type. So one of the pioneer of the root research, Professor Jonathan Lynch, with whom I did a short postdoc, he proposed that it is not always good to have too many roots. Rather, roots should be properly branched, and we should have root branches not only in topsoil but also in the deep soil. Plants should have more dispersed lateral roots rather than a bushy root system. Plants should have longer root hair, and root should also have the cortical parenchyma. And I'll show you example that the genotypes with such adaptive root traits, they perform better in phosphate deficient soils. Here is one example. This example was they have demonstrated this in the maize. If you see here, here is a root which is full of cortical cells. Now they are living cortical cells, and since they are living cortical cells, they need carbon for respiration, and they also need phosphorus. But in low phosphate condition, the maize root cortical cells, they are replaced by dead parenchyma cells. Now water and nutrients, they can easily come up to the endodermis by apoplastic mode of transport. So you really don't need these cells have to be a living cells. These root can afford the dead cortical cells. Now, when we have the dead cortical cells, we have essentially cut down on the carbon use, and we have also cut down on the phosphate use. And the genotype with more cortical parenchyma, they perform better in drought stress condition and also in the phosphate deficient conditions. So architectural change in the root anatomy can help improve phosphate uptake at the minimum carbon cost. In fact, we found the same thing is happening in rice. Here are the two cross sections of rice root using this laser ablation technology. This is a wonderful technology. This is available, I think, 2 places in the world. One is Nottingham University and another is the Penn State University in US. So here a precision laser is used to cut the sections of the root, and then computer can superimpose those images to give you a 3D picture of the root anatomy. You can literally walk inside each cell of the root. So since rice grows in a quite a watery condition, so rice roots naturally they have higher cortical parenchyma, but still you can see some of the living cortical cells. But in the phosphate deficiency, what we found that in fact in rice also almost all the cortical cells are now dead cells, and they are replaced by the large parenchyma cells. And although we could not test whether such variety can perform better in the phosphate deficiency, this we will come to know in the due course of time. But you take it from me that rice also respond to phosphate deficiency by killing the cortical cells in the root and replacing them with the parenchyma. And this adaptation is targeted towards saving the carbon cost of the soil exploration. Now, besides anatomical traits, there are several other traits. For example, root hair. Root hair are nothing but the epidermal cell elongations. So these epidermal cells, they elongate and they increase the root surface area tremendously. And this is also one of the adaptive trait which doesn't demand too much of carbon because there is no cell division. Here it is only cell elongation. And these tiny cell elongations, they can increase the root surface area tremendously. Not only this, root hair actually help in the seedling establishment during the germination. In if this can play, yeah. So here you have wild type germinating seedling, and if you see that when root is penetrating into the soil, the root hair are coming out and they are fixing root into the soil so that now the elongating root can push through the soil. So this is the role of root hair in the early seedling germination. Otherwise, if root hair are missing, as you see here, this is a root hair less mutant, and there is nothing behind root tip which can fix root into the soil, and therefore this seedling is having the difficulty in penetrating the soil, and most of the part will come out of the soil, and which will result in the poor plant growth. So root hair are also important for early seedling establishment. We also know that phosphate is not very much soluble in the soil, and most of the phosphate remains in the topsoil. So plants having a shallow root system, they theoretically they should absorb more of phosphorus. While, for example, nitrogen, which is highly soluble, especially the nitrates are extremely soluble in water, and a nitrate go down with the water deep into the soil, and plants prefer a deep root system in nitrogen for nitrogen uptake. Now, plant respond to phosphate deficiency also by elongating root hair. This is a root without any phosphate deficiency. Here we have a root with phosphate deficiency, and 80% of phosphate uptake is actually by root hair. Now, what if we combine these two traits in one plant? I mean, the shallow root system and longer root hair. And this has been achieved again by Professor Jonathan Lynch in US, where they have done it in the bean. And you see here, when the roots are shallow and they have longer root hair, the plants grow better in phosphate deficient situation, which is the first case, the longer root hair and shallow root system. What if we do it other way around? We have longer root hair, but a deep root system. The growth is poor, and other combinations. So this is a perfect example of root trait synergism. So all the traits, they may be antagonistic as well, but if we can identify root traits which are supporting each other or which are synergistic to each other, we can improve the plant performance in poor soil or in the phosphor plant or the soil which are not getting the proper phosphate fertilizers. So while these things were not known, what was not known that how this root angle, which determines whether the root would be shallow or deep, or how this root hair length, how they are regulated at molecular biology level, or what are the genes which are regulating such adaptations. So root angle is determined by root gravity response. Faster the gravity response, steeper the angle. Slower the gravity response, shallow is the angle. And gravity response is defined by two other factors. One is the differential oxygen accumulation. So when we give the gravity stimulus by simply rotating a germinating seedling by 90 degrees, so most of the oxygen moves into the proximal side, and where the higher oxygen concentration inhibits the cell expansion. While on the distal side, there is less oxygen, and these cells now will expand. So because of the differential expansion of cell, the restricted on the lower side and higher cell expansion on the upper side, root start bending towards the gravity. This response is also regulated by these amyloplasts or statoliths, which are there in the root tip, root columella cells. And during the gravity stimulus, these statoliths, they sediment towards the direction of gravity, and they actually give a kind of a physical force to bend the root towards the gravity. So coming back to the question that how phosphate deficiency regulates whether the root would be a shallow root system or a deep root system. So this work we did in the collaboration with Dabing Zhang in China and Malcolm Bennett in UK, where they identified a mutant, which is RMD mutant, root morphology determinant, and this mutant develop a steep root system. And there are really few roots at the shallow depth, and this mutant has a poor phosphate uptake efficiency. What we found that is RMD is low phosphate responsive, and cutting a very long story short, I can tell you that RMD protein is actually responsible for binding of actin filament with the statolith in the root columella cells. So with more filament binding to statolith, they are slowing down their sedimentation rate. And when their sedimentation rate is slowing down, the gravity response is slowing down. So when RMD levels are high, the slow sedimentation rate of statolith in columella cells will result in the shallow root system. But when in the high phosphate condition, when RMD is missing, the sedimentation rate of statolith would be faster and plants develop the steep root system. And let me tell you that these steep root system plants, they had poor phosphate uptake efficiency. So theoretically, the plants having the shallow root system, they should uptake more phosphorus. So we created a mutant in rice by disrupting the auxin transport, by disrupting this gene OX1. And these mutants, they do not respond to gravity. And as a result, the mutants plant, as you can see here, they develop a shallow root system. This is a normal plant, wild type plant, and these two are mutants. Now, since this is developing a shallow root system, ideally we thought now that this mutant should absorb more phosphorus. When we grew these mutants in the pipe filled with the high phosphate soil at the top and low phosphate soil at the bottom of the pipe. But to our surprise, these mutants did not absorb the more phosphorus compared to wild type plant. So we were a bit puzzled that why is this happening? And on closer look, what we found that while disrupting the root angle, unknowingly, we also disrupted root hair response. So rice also respond to phosphate deficiency by producing longer root hair, but these two mutants, they do not respond to phosphate deficiency by elongating root hair. So this is another example, and this is just proving that root angle alone is not responsible, but root angle along with root hair are important for phosphorus uptake. In the next paper, we gave the complete mechanistic of this response, and we proved that low phosphate responsive elongation needs concerted activities of auxin biosynthesis at the root tip. It transports across the lateral root and epidermal cells, and it responds in the root elongation zone where it enhances the length of root hair. So with this basic knowledge in hand, we were looking for a gene in rice which we can target for improving root hair length using transgenics, and we found a transcription factor which is a BHLH domain containing transcription factor. And when we overexpress this gene, we found the roots have not only longer root hair but also denser root hair. In addition, we found that transgenic lines in along with longer root hair, they also have the longer lateral roots. These are the root branches on the rice nodal roots. So we named this gene as a rice root system modulator 1, RSM1. And when we grew these transgenics in the phosphate poor soil, these transgenics not only accumulate more phosphorus, they also accumulate more biomass. Later on, we proved that this is happening because of the more auxin accumulation in the roots of overexpression plants, because RSM1 regulates the many auxin related genes. So this was the work we done in rice. We also did some work in chickpea, because relevance of chickpea, relevance of root hair in chickpea is twofold. First, they are important for nutrient uptake, but they're also important for nodule formation, because rhizobia enters through the root hair. So we wanted to see how chickpea respond to phosphate deficiency, and we found actually chickpea also respond to phosphate deficiency by elongating root hair. So we screened almost 211 chickpea genotypes for root hair growth under phosphate deficiency, and we ended up analyzing almost 5,000 images like this, and manually measuring the root hair length. So in a way, we actually measured 2 lakh root hair for their length. And we found that not only chickpea respond to phosphate deficiency by elongating root hair, there is considerable variation which is significant between the chickpea genotypes in terms of root hair elongation under phosphate deficiency. So we collaborated with Dr. Sarup Parida. He had SNP data, and we performed the GWAS and identified few significant SNPs. And what we found that those SNPs are actually falling in the genes which are homologue of which are known to regulate the regulate the root hair length under phosphate deficiency. So we proved that in one more crop that this root hair response is conserved to phosphate deficiency. So in the summary of this part, I would say that improving phosphate uptake roots are the key, because smart roots which are adapted to local soil type, they can enhance the phosphate uptake. I have shown you how RMD1 can regulate the root angle in phosphate deficiency dependent manner. Root hair are critical for phosphate uptake. So not only architecture, root functions like root secretion of acid phosphatase from root, or also root anatomy, like having the more root cortical parenchyma. All these things can help improving phosphate uptake. Now, let me switch to the second part of my talk, which is about phosphate use efficiency. While plenty of work has been done on phosphate uptake, you don't find many papers on phosphate use efficiency. Rice cells or any plant cells, they accumulate the insane amount of phosphate. And this gives us opportunity to improve phosphate use efficiency, because plants do not need that much phosphate. Almost 0.2% of plant drive mass is phosphate, which includes the inorganic form and organic form. And cytosol surprisingly has about 1 to 10 millimolar phosphate in the plant cell, which is really, really high when we have hardly 2 to 20 micromolar in cell. And 70% of this phosphate is stored in the vacuole. So in the event of phosphate deficiency, it is the vacuolar phosphate which is initially used to balance the cytosolic phosphate. So first there is a depletion of vacuolar phosphate, and when there is a depletion of cytosolic phosphate, then under severe phosphate condition, it is the recycling of phosphate from organic phosphate pool in the cell. Now, what are those organic phosphate pool? These are the biomolecules inside the cell, and they accumulate more than 50% of organic phosphate in the cell, like ATP, degrading RNA, degraded DNA, other membrane components. So all these biomolecules, they are recycled to release phosphate. And one such example is the replacement of phospholipids by galactolipids to release phosphate, because galactolipids do not contain phosphate and phospholipids contain phosphate. I'll come to this. So optimizing the recycling of these cellular organic resources can help us improve the phosphate use efficiency. But one of the problem is, I reiterate again, that we really do not know many genes which can be targeted for improving phosphate use efficiency. Now, before I go further, let's understand this. Here, what you see is the DAG. This is diacyl glycerol, simply a glycerol moiety, and 2 carbon chain, acyl chain. When this DAG is combined with sugar, that is galactose here, with the help of this enzyme, it becomes MGDG, monogalactosyl diacyl glycerol. That means one galactose combined with DAG. And this MGDG, if it is combined with one more galactose with the help of this enzyme DGD synthase, it becomes DGDG. That means digalactosyl diacyl glycerol, 2-DGDG. Now, if DAG combines with phosphate group instead of sugar, it becomes phosphatidic acid. So upper two, the MGDG and DGDG, these are galactolipids. These are lipids, but without phosphate. But phosphatidic acid and many other lipids, they are phospholipids. That means they contain phosphate. Now, MGDG, in 3D picture, it forms a cone-shaped structure, while DGDG, it forms a cylindrical shape structure. And I'll come to relevance of this. Here is the enlarged view of plasmamembrane and chloroplast. In plasmamembrane, most of the lipids are phospholipids. That means the phosphate-containing lipids. In chloroplast, the outer envelope has phospholipids and few galactolipids, the phosphate-free lipids. But the inner envelope has majority of galactolipids and some phospholipids. Now, these are the thylakoid membranes, the most abundant membrane on the earth. and they are highly stacked. So there are so many corners. Because of this structure, MGDG is the ideal lipid for galactolipids, for thylakoid membrane. And thylakoid membranes are entirely composed of galactolipids. That means thylakoid membranes are free of phospholipids. Okay, now what happens under phosphate deficiency? These phospholipids or P lipids are hydrolyzed. They are replaced by non-phosphorus lipid. Carbon acyl chain length is altered, and also the double bond index between the carbon is altered. These three changes, we collectively call them as membrane lipid remodeling. Phospholipids in membrane, they store about 30% of phosphate. And when they are degraded under, a portion of them are degraded under phosphate deficiency to release inorganic phosphate for cellular reactions. And these phospholipids are replaced by galactolipids, mainly DGDG, which has the cylindrical shape. But this process is reversible. When plants get phosphate, the phospholipids are back in the plasma membrane. And this is a well-conserved strategy in bacteria, and many of the plants which are tolerant to extreme phosphate deficiency, they can recycle almost 80% of their phospholipids. So we asked a question, what is the case in a fast-growing crop like rice? So to understand this, we screened large number of rice genotypes and did lipidome profiling of these rice, some of those lines. Here the data of 10 genotypes. This is the sufficient phosphate condition. Blue line is galactolipids and red is phospholipids. So in phosphate deficient condition, you see the level of galactolipids has increased and the level of phospholipid has decreased, which is expected. And this means that this response is also conserved in the rice. And this is a very interesting adaptation because it can be targeted since it doesn't demand any phosphate from inside. It is using whatever phosphate is there inside the plant. But the problem is that what are the genes involved in this response under phosphate deficiency and how those genes are regulated. So we have identified few genes of this pathway of galactolipid phospholipid hydrolysis. And this is one of the gene GDPD, which encodes the glycerophosphodiester phosphodiesterase. This can hydrolyze the glycerophosphocholine, which is a phospholipid, to the glycerol 3-phosphate, which is then subsequently hydrolyzed to release phosphate. So we identified one GDPD gene, which is GDPD2, and it is highly responsive to phosphate deficiency. Its overexpression lines, they showed a greater level of repeat remodeling under phosphate deficiency. And these lines also had longer root length. And as a result, under phosphate deficiency, the GDPD2 overexpressing lines, they accumulate more phosphorus and they are tolerant to phosphate deficiency. So this is one example where the hydrolysis of enhanced hydrolysis of phospholipids can improve tolerance to phosphate deficiency. Recently, we started working on another gene which is involved in biosynthesis of galactolipids. So earlier example was on hydrolysis of phospholipid. This is on the biosynthesis of galactolipids. And this is a MGD synthase, which synthesizes the MGDG. There are almost 3 MGDGs because MGDG is essential for chloroplast functions. So MGDG is always needed. But this gene MGD3 is not always expressing, it is low phosphate inducible. So this gene is actually involved in the MGDG biosynthesis or galactolipid biosynthesis when there is a phosphate deficiency. And we found that it responds exclusively to the phosphate deficiency, doesn't respond to other mineral deficiency. We also found it's a chloroplast localized protein since MGDG synthesis occurs in the chloroplast. We also found that this gene is regulated by PHR2 transcription factor, the one which regulates almost everything. In the PHR mutant, induction of MGD3 is minimum, and using EMSA, we demonstrated that PHR2 actually binds to the promoter region of MGD3. So to further understand its function, we used CRISPR-Cas9 and we generated the loss of function mutant of MGD3, and we got a very high mutation frequency of 88%. And from many mutants, we selected few mutants, which I'm highlighting here, with single base pair change. In next generation, we self these lines and identified the homozygous mutation. Homozygous mutations means the same mutation, same base change on both the allele of the MGD3. And by selecting the alleles where there was only single base addition, which was leading to the stop codon, we got homozygous lines. And next, we thought that since Cas9 is a protein which can target the off targets and which can target the other closely related genes, so it is good to get rid of Cas9. So we thought of getting a marker-free line, the tDNA-free line, in next generation. And this CRISPR-Cas9 system, it's a beautiful system to get marker-free lines because the site of tDNA integration is quite far away from the Cas9 activation site. So we have the site of mutation separated by hundreds of kilobase pair of DNA from the tDNA integration. So in the next generation, these two loci, they will segregate independently, and this gives us a chance to identify a line which is tDNA free but mutated. So we can also get a marker-free line by using tDNA free gene editing right in the beginning, where we can use artificially synthesized guide RNA or RNP or the Cas9 from the bacterial proteins. We can coat them using particle gun. We can bombard the rice calli. And here the benefit is that Cas9 protein and guide RNA, they would be transiently stable. There would be no integration in the plant genome. And therefore, chances of off target or any carryover to next generation is almost impossible. But this system is a poor efficiency. We hardly get 10 plants using this tDNA-free RNP technique. So we went back to the this segregation strategy. Here key is that if you identified a single copy line, the line which has only one tDNA integration, but mutated or edited. So in the next generation, these lines will segregate 1:2:1 genotypically. So idea is to find this plant, which is a null segregant for tDNA, but edited. And this can be easily identified using PCR. As you can see here, we identified that there is no tDNA when we were doing PCR for tDNA, but this gene was edited. So by this way, we identified a T3 lines, which is completely marker-free, and there is just one base pair change in the gene, which is leading to stop codon or complete loss of function. So this mutant is as good as the EMS-generated mutant. So using this line, we found that knockout lines, they showed poor galactolipid or reduced galactolipid accumulations. And when we tested the phosphate use efficiency under -P condition, mutant performed poor. So that basically indicating that MGD3 contributes toward the phosphate use efficiency in rice. We validated this using overexpression lines. Overexpression lines had the higher galactolipid accumulation, and they also had higher phosphate use efficiency. So this is an interesting strategy where actually plants are not demanding phosphate from outside, rather they are prioritizing the use of phosphate which is already present in the plant. So with these examples that there is lipid remodeling in the rice and they are the genes which can be targeted, we thought of another question. I was interested in understanding the evolution of such pathways or the adaptation of this response. And my question was that, do natural rice genotypes show variation, significant variation in lipidome changes under phosphate deficiency? If it is true, if different rice genotypes or diverse rice genotypes which are having the variation in phosphate deficiency tolerance, do they also show significant variation in lipidome change? That means the hydrolysis of phospholipids and accumulation of galactolipids. So to understand this, we screened a large number of rice genotypes and we divided them into tolerant and sensitive bulk based on PUE, phosphate use efficiency. And we performed comprehensive lipid profiling. And if you see here, in normal condition, galactolipid level are about 65%, and in -P condition, their level zoom to almost 90%. While phospholipid level is about 35%, but in -P condition, their levels are reduced to less than 10%. So this is telling me one thing, that this response is conserved in rice. All the varieties, irrespective of tolerant or sensitive nature, they are showing the lipidome changes, enhanced degradation of phospholipid and enhanced synthesis of galactolipids. But from this picture, we do not tell whether there is a significant difference between tolerant and sensitive. Now, when we did statistical analysis of this data, we found that tolerant or sensitive genotype or high PPUE or low PPU genotypes, they did not show any difference in the percentage of phospholipids and or in the percentage of galactolipids. So this was first unexpected data we got. So then we dig further into our data. As I said, this is a comprehensive data. So we have the molecular level data of these rice genotypes. So we analyzed what is happening to DG level, the individual lipid classes, MG-DG level, and many phospholipid classes, phosphatidylcholine, phosphatidylethanolamine, phosphatidylglycol, and phosphatidylserine. Here also, we did not see a pattern. There was no difference, clear-cut difference, or no significant variation between the tolerant bulk and the sensitive bulk. And this was really, really unexpected. I was expecting that there would be some changes, but no change in the total lipid, no change in the major lipid classes. We dig it further, and then we analyze data at the individual lipid species level. So MG-DG is composed of many MG-DG species, which may show difference in the carbon chain length in the double bond index, and because of that, they can be classified differently. So when we went to individual species level, we found a clear-cut significant difference between tolerant and sensitive bulk in 3 DG species. Here, the 34 is the total carbon number and 4 is the double bond index. And we found the clear-cut association of, not association, the correlation of the phosphate use efficiency with 3 DG species and 1 DG species. So sometimes when you don't get the obvious or expected results, you discover something new. And there was one report before our paper that was done in some other group. I can't recall it right now. And second report was from our paper in rice, where we have demonstrated that these lipid species also play a very important role. So in the summary of this part, as I've shown you, that repeat remodeling under phosphate deficiency is a conserved and adaptive response in rice. Both tolerant and sensitive genotypes, they have displayed a similar level of lipid change under phosphate deficiency. However, there are significant differences in the galactolipid species. Now, these species, they are reported to have signaling roles in humans and in other animals. But in plants, I could not find a paper where somebody has demonstrated their roles. And that means this leaves an area where we are going to work further, that these species might have signaling role, which is playing helpful under phosphate deficiency. As I said, that lipid signaling is extremely poorly understood in rice, and we are going to investigate it further. But nevertheless, the genes involved in repeat remodeling, they can certainly be targeted for improving phosphate use efficiency. And I say it again, that is a very attractive target for improving PUE, as it does not demand the extra phosphate from outside. So coming to the last slide, which is the way forward. So what are the targets we have, which we can target for improving either phosphate uptake or its use? Roots, the most attracting or the excellent trait. But we really don't understand much about rice adaptation to phosphate deficiency, rice root adaptation to phosphate deficiency. There are almost 25 quantifiable traits in the root architecture, and the land races, wild relatives, which are naturally tolerant to phosphate deficiency, they should be explored to understand the adaptive root architecture in different soil types. So this is one of the obvious targets which should be targeted for improving phosphate uptake efficiency. Root microbial association is used extensively by trees for especially the fungal association, where fungal help in the phosphate solubilization. Many crops also showed very strong root microbial association for phosphate uptake. It is not really well understood in rice, and it could be because rice mainly grows in muddy soil, anaerobic condition, where the microbial association is not that easy. But it is certainly useful in the upland rice, and there are few reports where some of the fungal association actually help in the uptake of phosphate transport in rice by activating few transporters. So this is another area which we should look forward to work. Another important target is reducing seed phosphate content. When crop is harvested, the seed means taking away phosphate from the soil. And it is demonstrated by Fangma group beautifully that rice seeds, which have the high phytate content, quite a high phytate content in the rice bran, it can be reduced by 50%. So if you reduce the rice phytate content, which is essentially phosphate, by 50%, there is no compromise on the seed germination in fertilized soil. So this is one area where we can reduce the phosphate removal from the soil. Also, the new target should be identified for improving phosphate use efficiency. We really do not know much. The only target which we are working, or many people are also working, is this phospholipid remodeling, membrane repeat remodeling. But there can be many other targets. Phosphate is there in many biomolecules. In the RNA, degrading RNA, ribosomal RNA has the 80% of cellular phosphate in the cell. So those are the different targets which should be explored for improving phosphate use efficiency. With this, I must end. And before that, I must thank people who are working in my lab. This lipid work was entirely done by Lokesh, who is now postdoc in my lab. Pavandeep is working in the root areas, and other people are working in different areas. I must thank funding from DBT and my own institute. And some other time, probably we will discuss the project we are doing with Sundaram sir, where we are trying to identify QTL, which is not PUP1, but other QTL in rice for improving uptake and also use efficiency. Thank you very much. I would be happy to answer any question if I can.
Many thanks, Dr. Jitender. On the lighter side, I couldn't recognize you from the photograph of students. You still look like a student only.
Thank you.
It was a wonderful lecture, starting with the basic mechanisms of P uptake and transport into different cellular parts. You gradually graduated into discussion about the role of root hairs and the role in phosphate acquisition and utilization. And we also heard about some of the exciting developments related to lipid remodeling, which can enhance phosphorus use efficiency. Overall, I would rate it as one of the best presentations we had under Azadi Ka Amrit Mahotsav on part of both Society for Advancement of Rice Research and also on part of IARR. Dr. Jitender, before we open the house for questioning, pardon me if I'm a little bit naive. I have a few queries. The first question is, you mentioned that OSPH01 is possibly associated with transport of phosphorus from various other plant parts into seeds. Am I right?
So FOR1, sir, actually FOR1 is a very interesting transporter. I have actually two projects on this gene, single gene. It differs from the classical transporters. So its first job, as it was discovered by Yves Parrier group in Switzerland, its first job is that it expresses somewhere in endodermis. And it loads the phosphate from the root cortical cells to the root xylem, from where it can go along with the transpirational stream to the upper part. Recently, Fangma group has shown that FOR1 also loads phosphate into the developing seed. This is just, I think, 6 months or 1 year back, they published in Nature Genetics or some paper. So FOR1 is, this is a new discovery where it not only load phosphate in the root, but also in the seed.
Is it like, say, how many copies of genes encoding FOR1 are there in rice?
There are 3 in the rice, and FOR1, FOR1/2, that is the main gene.
Okay. So my question is, if FOR1 or FOR1-2 has a very critical role in phosphate loading in seed, one of the major obstacles wherein we are facing, not only in rice, many other crops like maize, pearl millet, and also wheat, is with regard to biofortification. As you are very much aware, phytic acid interferes with absorption of micronutrients. Is there any way to manipulate, either by knocking down or knocking out genes like OS41 or FOR1-2, and then lessen the phytic acid concentration?
Yes, sir. Yes, this is what I was mentioning actually. Fang Ma Group, some of you may be aware, he is working in Japan, and he is the person who has discovered this thing. So he has identified a transporter earlier, which was not annotated as a sulfur transporter something, but it actually transports phosphate, and it's a node-based transporter. So it expresses at the node of flag leaf, a leaf just close to the panicle. If we mutate that transporter, the transfer of phosphate from shoot part to seed is reduced by 50%. And he has demonstrated that in a fertilized farmer's field, that reduction in seed phytate is not affecting seed germination. Actually, the phytate is also needed, because if we have the free phosphate in the seed, then it affect the starch biosynthesis. Free phosphate actually inhibits the starch biosynthesis genes, I read somewhere. So that is the one transporter which has been demonstrated so far. In fact, I have mutants, several kind of mutant of CRISPR mutant we have generated for FOR1, and I did not see much change in seed.
Okay.
So they are just normal. We have to do more analysis if something is actually happening.
It will also be exciting or interesting to see allelic variation among rice germplasm, specifically with regard to FOR1 or FOR1-2, and then correlate that with their performance under high or low phosphorus conditions. So anyway, like say, we have at our disposal about 653K rice germplasm, and then we have already grown them for a single season during Kharif 2021. And we will certainly share the results with you. And you can analyze the allelic status with regard to FOR1 or its regulatory motifs. And then we can see if there is any correlation with regard to allelic variation with respect to FOR1.
Sure.
And the second thing is, has anybody studied the allelic variation with regard to OSMGD3, which appears to be the key gene with lipid remodeling?
No, sir, nobody has done this. In fact, I'm expecting, few things in the area that we are working, I'm expecting somebody is going to put them in some QTL.
That could be, that is possible.
Exactly, because,
And regarding the role of microbes, when you mentioned that under typical submerged conditions, microbes, particularly, I agree with you in one sense that fungal, like say fungi may have a minimal role because it's not totally an anaerobic condition. In rice, what we typically call as microaerophilic condition, there are a large array of microbes, including phosphorus solubilizing bacteria, which are indeed, my colleague Dr. Lata may throw more light in this particular regard. But there are a large group of phosphate solubilizing bacteria, which are also available commercially, which are indeed playing a major role. But whether they are playing a major role just in solubilizing the phosphorus, or do they have any other function? And are there any mycorrhizal association in a typical submerged rice field? These are all open questions, and then I agree with you.
Yes.
That is something wherein we need to put more of our efforts in terms of understanding microbial role with regard to uptake of phosphorus. That is something we need to work out.
Yes, sir.
Yeah. Thank you, Dr. Jitendra. It was a wonderful presentation. I now request Dr. Satyendra to open the house for questioning by others also. First, maybe you can read out, Dr. Ananta has, I suggest that people can raise their hand to ask questions, or type the questions in chat box. Dr. Satyendra, take over.
And also, please feel free to guide me. I may not be, right.
Dr. Satyendra, you have to unmute yourself. Dr. Ananta, you can go ahead.
Sir, Dr. Giri, so it was a really wonderful presentation, sir, as our director told. So last time also we listened to your talk, and this time also very informative and entirely new one, so we could know. So my question is, so we are targeting use efficiency and acquisition efficiency as explained by you in a different mechanism are involved. So if you want to improve both together in a variety, susceptible variety, so what are the genes we can target, or is it possible to combine these two mechanisms in a single genotype or in a single way? Can you please brief me?
Yeah, certainly, yes. I doubt that any plant is exclusively dependent on uptake. All plants, there's always a combination of both PUE and P uptake efficiency. And as I said that for uptake, many genes are known. Any gene which is improving root surface area will increase the uptake. Many root-bound phosphate transporters, people have demonstrated that overexpression of them can increase the phosphate uptake. But where we are lacking is on the PUE genes. And PUE genes, I really don't see any, kind of a significant gene which can be directly targeted. This MGD3 or other genes, they are overexpression based approach. To find an approach, we need what Sundaram San has suggested. We need to look for the natural allele, the allelic variation, which may be more functional or more, active, biologically active, so that they can synthesize more galactolipids. But as of now, honestly speaking, I'm not aware of any gene which can be targeted using breeding approach for PUE. If anybody in audience, if anybody has any suggestion, I would be happy to take that.
Dr. Satyendra, kindly unmute yourself and then go
Yes,
ahead.
sir. So in the chat box, we have a question from Dr. DVK Nageswara, sir. His question is that there must be some threshold for the plants to respond to induce the tolerance to low P, which must be defined as why and how it is low. If so, what is the quantitative level of P outside the root system that triggers reactions in plant system, for instance, in rice?
All right. Yeah.
Yeah.
All right. So I'll answer the second question first. So, sir, there are two responses in plants. One for phosphate, one is called systemic response and another is local response. Systemic response is that when plants cytosol start feeling phosphate deficiency. So there is a systemic response. What if I have grown a seedling in a perfect phosphate fertilized media and I transfer that seedling to A minus P media? Now here in this case, seedling has plenty of phosphate, but within 3-4 hours, you will see the induction of phosphate deficiency response. Now this is local response, which is not dependent on the phosphate P status, rather dependent on the rhizosphere P status. So there is no threshold for the local response. Moment it sees that it is not, the phosphate is not there, it will start responding. Now coming to the that there is some threshold for phosphate level. I doubt anybody has defined this, but usually every paper will see the different phosphate level. But in the science paper by Wild et al. 2016, when they discovered that inositol 8-phosphate is actually used as a sensor. So they have defined the binding of SPX protein with IP8, and there they have done titration, if I remember correctly, titration, and they've calculated the Km values, and they have defined the concentration of phosphate at which IP8 can bind or it cannot bind. I can't recall the exact concentration, but this is defined in cellular context. But in the whole plant level, I think it's not possible to define. It will vary with the soil type, and it will vary with the plant stage. A young plant may feel more deficiency compared to the germinating seedling, which will derive phosphate from seed itself.
I would like to supplement what you had mentioned, Dr. Jitendra. Agronomically speaking, we consider any levels that are lower than 10 ppm as moderately low, and any levels less than 5 ppm as very low. I hope I'm using the right terminology here, Dr. DVK. Okay. Satendra, are there
Yes,
any...
sir. So we have more compliments for Jitendra than the questions in this presentation. So Jitendra, one question is from Dr. Pradeep Sharma. Does the same mechanism happens in wheat, also being cereals?
Dr. Pradeep Sharma, I would like to introduce. He is one of the very, like say, young scientists who is doing active work in wheat biotechnology. I think you should get in touch with him, or Dr. Sharma should get in touch with you to explore more such opportunities for collaboration in wheat also.
Yeah,
and his another question linked to this is, how about other modifiers in plants, say small RNA, while altering the gene of interest?
Yeah, I mean, this is conserved in wheat because of polyploid. Not many people have studied it, but I'm sure it's pretty conserved, this response. Regarding small RNA, microRNA 399, it is one of the key regulator of phosphate deficiency response. Then there is IPS1. That's another small RNA, which is also the signaling molecules for, though both of them are involved in the feedback regulations. They regulate the phosphate transporters, ubiquitination of phosphate transporters by FOR2, sorry, the FOR2 is a ubiquitin conjugase. So those small RNA, they are, I mean, by the target mimicking, they regulate the phosphate response.
Carrying forward with this particular suggestion, will there be a role for epigenetic modifications like methylation or acylation in terms of?
Yes. This is also been worked out by one of the student of Professor Raghutama, who is also one of the pioneer of phosphate research in Purdue University. I think Aaron P. Smith, he's working in this area. There is certainly epigenetic regulation.
Thank you.
Yeah, Dr. Raghu's lab they were working on. Yeah. We have another question. I think this you have already addressed. Even then, I will read out the question from Dr. Sunil. Whether root length will enhance the phosphorus uptake in rice, or the genotypes with more root biomass and longer root hair length will enhance it. So
yeah, again, as I'm repeating, I mean, these are not the absolute statements that if you have longer root hair, it will certainly increase the phosphate uptake. They are all dependent, condition dependent. They depends on many other things. For example, I showed that where root hair are located, something called straight synergism. If root length is deep and root hair are longer, they are not of much use, as is demonstrated in bean. But if the root hair are located on the root branches which are close to the surface, then they are beneficial.
yeah, and then Madam Padma Kumari has a question or a suggestion. Phosphorus as a major fertilizer is recommended for basal application in rice field. So with the available knowledge on phosphorus use efficiency, can we bring about a change in the time and the level of application in the field for optimum uptake?
I think this question some agronomists should answer. This is not something for, but anyway, here I can.
But it's a thought-provoking question, I would say.
Yeah, yes. So here I can add one thing, sir, because this nanotechnology or nanophosphate particles. Satendra, you remember Rahul has published one paper recently on this. So they have prepared the nanoparticles for phosphate, and they have demonstrated that it can increase the use efficiency.
Yeah, in fact, Rahul, he collaborated with our institute, and Dr. Gobinath, I remember, was working with Dr. Rahul to demonstrate the efficacy of nanophosphorus in rice. Yeah. So, yeah, maybe Then we have Sakshi, sir, is it okay to get no or minimal root hairs in rice plants when grown in low P, that is 2 to 5 ppm? Maybe if Sakshi you are there, maybe you can elaborate a little bit more and make the question more clear. Then we have Priyanka. Sir, can you please elaborate about the root diameter and the number of root crossings role in phosphorus use efficiency in brassica?
Yeah, this is interesting question actually. Thicker roots, they are considered anciently old roots. And the new concept is that plants should have thin, fine roots, because thick root means the many layers of cortex, which will add to the carbon cost. If you have the fine roots, we will find thin roots, they can penetrate the small pores in the soil and explore more area at the same carbon cost. So root diameter is another trait, which is a very important root architectural trait. And I think it is demonstrated in nitrogen use efficiency again by Jonathan Lynch. I will recommend you that you read some of the papers from Jonathan Lynch lab. Recently, he wrote in Annals in Botany, he wrote a review where he had described the relevance of many root traits like root diameter or many other traits. But yes, root thickness, thicker the root is not considered, a fertilizer efficient, at least in rice.
Yeah. In fact, now there are no questions. I have something to compliment Dr. Jitendra. So recently, we worked on understanding the seed phosphorus level on germination using Nagina 22 mutants. We used large number of mutants, and we have identified 2 or 3 such mutants which accumulate low phosphorus in grains, but they have equally, good seedling vigor. The paper will be probably out in scientific reports in 1 or 2 days. It will be out, wherein we have done thorough characterization of a large number of mutants and trying to understand that can we reduce the level of phosphorus in seeds. And there are few mutants which they accumulate less phosphorus without any adverse effects on germination or seedling vigor. So maybe those are very good material for breeding and also breeding for low grain phosphorus and also for basic studies.
Yeah, agree.
Yeah. Dr. Jitendra, there are a few more questions. Are you ready to take them, Dr. Jitendra? Or
this is Saturday afternoon. I am alone in my lab, so I can stay here as long as you want.
So I have one question, Dr. Jitendra, before we move to other questions. We have certain rice varieties like Moraberakan, which are all, etc., belong to tropical Japonica group. They have very profuse root system, and they are known to be drought tolerant, but they are not indeed tolerant to low soil phosphorus. So the hypothesis that a deeper or a more branched root system may be associated with tolerance to low soil phosphorus, how true is the hypothesis, particularly in case of Moraberakan?
So as I said, that these are not the absolute truth. It has been reported in few cases and it is working fine and it appears theoretically correct. But again, as I said, that there are many other factors. Soil is a very complex environment. So there are many other factors, and unless you test them in your local soil type, we cannot comment on it. We can theoretically speculate that it must behave better. For example, we were expecting in case of our OX1 mutant, because of shallow root system, it will absorb more phosphorus. And for 3 years, we were struggling why it is not showing phosphate uptake. But somehow then something it clicked that, oh, it could be root hair. So there could be many more things, sir, which may influence. So unless you test in the local soil type or the soil where you want to study them, it is difficult to tell for guaranteed. But theoretically, a shallow root system is better for phosphate uptake in natural environment.
That's fine.
But then it may be a negative thing for water uptake.
There is a trade-off between phosphate uptake and water uptake. There is a trade-off.
Yeah. And
So
we need to remember that PUP1 works both under water limiting conditions and also
Yes.
under low soil phosphorus conditions.
Yes.
Satyendra, maybe a few more questions are there. You can read them.
Sir,
out.
yeah, I'll read out, sir. Madam Padma Kumari, what is the stage of the plant you use for all your P efficiency studies? So,
Madam, these are 1 month old plant in hydroponics.
okay. Then Dr. Sunil, if we cultivate rice in excess allocation carbon soil and the rice genotypes have longer root hairs, it may enhance phosphorus uptake. That is his question. And is it possible? Why I am asking this is excess allocation of carbon to root may suppress the shoot growth. Yeah, I could not understand the question properly.
Yeah, one thing I can clarify that carbon in soil is like a party for microbes. So microbes will never allow plants to take carbon from soil. Plants are not designed in that way. Plant will always get carbon from air, and microbes will always get carbon from soil. So if it is about the carbon supply in the soil, I doubt it's going to benefit plant at all. It will attract more and more microbes only. Yeah, but I'm not clear what he exactly wanted to ask.
Okay, Sakshi from ICAR-IIRR. Maybe Dr. Sunil, if you can, you can unmute and clarify the question, that will help us. Then we have a question from Sakshi ICAR-IIRR that we have grown rice seedlings in hydroponics at 2 ppm concentration, and we observed that many of the plants have no root hairs or minimal root hairs as compared to control.
So if you read the recent paper from Malcolm Bennett group in the Science, plants sense the space for branching. So in hydroponics, actually hydroponics is not a very good system for studying root hair because there is absolutely no space, no air. So plants sense that hydroponic system as a homogeneous system. But if you grow the same seedlings on the media or in some soil, you will certainly see the root hair elongation.
Yeah, I think the next question probably we have discussed. Any relation with the phosphorus use efficiency and water use efficiency? By Dr.
That's a trade-off. It's a trade-off.
Yeah. And yeah, I think with this, if there are any more questions from the
Dr. Mahindra Kumar, he's our General Secretary of Society for Advancement of Rice Research. He's also heading our agronomy section here, and he has active interest in both water saving technologies and also low P tolerance. He has a question, is there any relation between phosphorus use efficiency and then water use efficiency? I think this is a very good question. Sir, yeah. I understand that Dr. Mahinder is on tour. Somewhere in the car, he's managing to hear the proceedings and then ask these questions. So,
yes, so we appreciate commitment. He is attending it. Yeah. So, Dr. Mahinder, as I said, that between phosphate uptake and water uptake, there is a trade-off. Shallow root system and a deep root system. But again, I'm saying that these are not the absolute truth. These are based on few studies and few papers. It doesn't mean that shallow root means that all the roots are close to the surface. We will also have some roots which are deep into the soil. And if you ask me molecular level, let me tell you that phosphate deficiency also affect water uptake. There is not many paper, but since you asked me, so it occurred to me. There are aquaporins, which are water channel proteins, and it is seen that phosphate deficiency shut the aquaporins. So water exchange is inhibited in the presence of phosphorus deficiency because aquaporins opening and closing is regulated by phosphorylation. So when phosphorylation is low, their closing and opening is not regulated that systematically as it should have been. So there are reports, yes, that phosphate deficiency may also impact water uptake.
Dr. Satyendra, are there any more questions?
No, sir. We are done with all the questions in the chat box.
I now request Dr. C.H. Padmavati, who is the Chief Editor of Journal of Rice Research, which is our society's journal, to kindly offer a vote of thanks.
Thank you, sir. Thank you. Am I audible, sir?
Yeah. Go ahead, ma'am.
Okay. Thank you, sir. Good evening to all of you. And on behalf of Indian Institute of Rice Research and also on behalf of Society for the Advancement of Rice Research and the whole rice fraternity, almost, sir, there are around 60 participants in this webinar today. On behalf of all of them, I extend sincere thanks to today's distinguished speaker, Dr. Jitendra Giri, sir, for making an excellent presentation, and your insights, particularly about the morphological and also the molecular biology insights into the root rights, are very exemplary, sir. Thank you very much, sir. Thank you very much for accepting our invitation and taking time out for this webinar. Thanks a lot. I thank our beloved and most dynamic director, Dr. R.M. Sundaram, sir, for taking keen interest on this topic and also for extending support for organizing this webinar. I thank the seminar organizing committee led by Dr. A.P. Padmakumari, Dr. Satyendra Mangrutia, and Dr. B. Shailaja, and the entire team who have taken efforts in organizing this webinar today. And I thank AKMU team led by Dr. Shailaja and her entire team for creating and providing links, and also for the uninterrupted streaming of this webinar. There is also a live stream that is going on about this webinar, sir. And I thank the SAR team, particularly the President Sundaram, sir, and also the General Secretary, Dr. Mahendra Kumar, for organizing, for being a co-host of this webinar. I thank all the participants, almost as I told, up till now for the presentation, there were around 63 participants who have taken, who were present here, for taking their time out and also attending this webinar, and also for the very lively interaction. And thank you very much, sir. I thank each and every one of you, and see you soon again in yet another event. Please stay safe and stay healthy. Thank you.
Thank you, Dr. Padmavati. If you kindly permit me, we are awarding this particular memento on behalf of Society of Advancement of Rice Research virtually now to Dr. Jitendra. Whenever we'll be that Delhi, or whenever you are here, this we'll be awarding it to you officially. Congratulations.
Thank you very much.
Thank you.
Thank you, sir.
And I must thank sir and others to arrange such a lively and wonderful audience on a Saturday afternoon. So thank you very much, sir. It was really nice talking to you people, and it was a chance for me to learn few things from you, the people who are working on the fact. Thank you, sir.
Thank you very much, Jitendra. Thank you.
Kindly switch on videos for the photographs. I request all
the participants to switch on their video or their photographs just to take a snapshot or record your presence. Dr. Ananta, I think you can open your webcam. Dr. Jitendra, many of our staff are down with mild or asymptomatic COVID-19, probably Omicron, including Dr. Ananta. And still, like say, even though they are in isolation and they are sitting, I appreciate their interest in terms of attending this particular meeting.
Dr. Ananta, please get well soon.
Thank you, sir.
He has been put in a jail at his home, I think.
Yeah, yes.
Yes, sir. Home
he doesn't have any symptoms.
Yeah,
many of us, when we tested, some of us were turned out to be positive.
So
but it's very mild.
Same is happening in Delhi. Many people are positive, even people are not getting themselves tested. So in a way, it's better. It doesn't, create a panic or something.
Ananta, get well soon. We need you back.
Thank you, sir. Most probably by Monday, I will get once again checked, sir. If it is negative, I will be through, sir.
Yeah.
But as such, no problems, no symptoms or any other things. Everything is fine.
Is it done, ma'am? Shailaja, ma'am?
Yeah.
Right.
All right. Thank you. Thank you, everyone.
Thank you very much. The meeting is finished. Thank you.
Have a nice
Thank you,
weekend.
sir. Thank you, sir.
And